Deep in the sun-scorched hills of Europe, a biological crime is unfolding that defies every rule we thought we knew. A species of harvester ant is doing the impossible, giving birth to a completely different species. Not hybrids, not mutations, but fully formed members of another ant lineage that split away 5 million years ago. These queens are cloning the males of their rivals, then using them as breeding stock to build armies of workers. It sounds like science fiction, like something from an alien world where the laws of nature bend and break. But this is real, happening right now beneath our feet. Tonight, we dive into one of the strangest discoveries in modern biology, a story of genetic theft, evolutionary genius, and the blurred line between species. The fundamental law. For as long as humans have studied life, one rule has seemed unbreakable. Species produce their own kind. Lions give birth to lions, oak trees grow from oak seeds, and ants raise more ants of their own species. This principle forms the bedrock of biology, the foundation upon which we classify and understand the natural world. It is so fundamental that we rarely question it, so obvious that it seems written into the fabric of existence itself. But nature has a way of humbling us, of revealing exceptions that shatter our assumptions and force us to rethink everything. In the world of insects, where life cycles twist into forms almost unimaginable, the rules we take for granted begin to bend. Some species produce offspring that look wildly different from their parents, shaped by seasons, population density, or social roles within a colony. Yet even in these cases, the offspring remain genetically part of the same species, bound by invisible threads of shared DNA. Until now, no one had documented a case where a species could give birth to members of an entirely different species. The very idea seems to violate the laws of biology, to cross a line that evolution itself should not permit. And yet, in a groundbreaking paper published in the journal Nature, scientists have revealed exactly that: a harvester ant species that lays eggs hatching into a completely separate ant species. This is not a story of close relatives blurring together, not a tale of recent divergence or subtle genetic drift. The two species involved, Messor Ibericus and Messor Structor, split apart more than 5 million years ago. In evolutionary terms, they are distant cousins, separated by vast stretches of time and countless generations. And yet, Messor Ibericus queens are producing offspring that are genetically and anatomically undeniably Messor Structor. The discovery began with a mystery that puzzled researchers across Europe. Colonies of Messor Ibericus containing hybrid worker ants in regions where Messor Structor did not exist. How could hybrids appear without the presence of the other parent species? The answer, when it finally emerged, was more bizarre than anyone could have imagined. The island without Structor, Sicily, the jewel of the Mediterranean, became the stage for this biological detective story. The Italian island is home to thriving colonies of Messor Ibericus, bustling with activity as workers harvest seeds and tend their underground nests. But something was wrong, something that didn't fit the pattern scientists expected to see. Within these Ibericus colonies, researchers found hybrid worker ants, individuals carrying genetic material from both Messor Ibericus and Messor Structor. This alone was not shocking. Hybridization occurs in nature when two species interbreed. But here's the twist: Messor Structor colonies are completely absent from Sicily. Not rare, not hidden in remote corners of the island, but entirely non-existent. How could hybrid workers exist without the presence of one parent species? The question hung in the air like smoke, defying easy answers. Biologists began to investigate, examining colony after colony, searching for clues hidden in the genetic code of these mysterious ants. What they found was astonishing. Within the Ibericus colonies lived two distinct types of male ants, visibly different from one another. Around 56% of the males were nearly hairless, their bodies smooth and sleek. The remaining 44% were covered in dense coats of hair, giving them a fuzzy, almost woolly appearance. When researchers sequenced the nuclear genomes of these different male forms, the truth began to emerge. The hairy males belonged to Messor Ibericus, as expected, but the hairless males had genomes matching Messor's Structor perfectly. These were not Ibericus males with unusual appearances. They were Structor males, living and breeding within Ibericus colonies. Yet no Messor Structor females were present anywhere in these colonies. No queens, no workers, no trace of female Structor DNA. The only logical conclusion was impossible. These Structor males must have hatched from eggs laid by Ibericus queens. The genetic proof. Science demands evidence, and extraordinary claims require extraordinary proof. The researchers knew they needed to confirm beyond any doubt that Ibericus queens were producing Structor males. They turned to one of the most powerful tools in modern biology, mitochondrial DNA. Mitochondria, the tiny powerhouses inside every cell, carry their own separate genomes distinct from the DNA in the nucleus. And crucially, mitochondrial DNA is always inherited from the mother, passed down through the maternal line without exception. This makes it a perfect genetic fingerprint for tracing ancestry. When the scientists sequenced the mitochondrial genomes of the hairless Structor males, they found something remarkable. These males carried mitochondrial DNA matching their Ibericus nestmates, even though their nuclear genomes belonged to a different species entirely. It was as if they had been born with two identities, Structor in their nuclei, but Ibericus in their mitochondria. In contrast, Structor males living in their own species colonies showed no such mismatch. Their nuclear and mitochondrial DNA aligned perfectly as nature intended. The mismatch was unique to Structor males inhabiting Ibericus colonies, a smoking gun pointing directly to cross-species reproduction. But the researchers didn't stop there. They needed more evidence, more layers of proof to support their extraordinary claim. They turned to the eggs themselves, examining larvae from Ibericus colonies raised in controlled laboratory conditions. Genetic testing revealed that 11.5% of the eggs had nuclear genomes matching Messor Structor. To confirm that queens, not workers, were producing these males, the researchers isolated 16 Ibericus queens and tested the genomes of every egg they laid. 9% had Structor DNA, while eggs laid by workers were 100% Ibericus. The queens were the source. The final piece of evidence came from direct observation, the gold standard of scientific proof. A laboratory colony containing a single Ibericus queen was monitored continuously for 18 months. Researchers watched, documented, and recorded as reproductive adult males of both species hatched from eggs laid by that single queen. The case was closed, the evidence overwhelming and undeniable. Messor Ibericus queens were cloning males of another species. The domesticated genome. This discovery is more than just a biological curiosity. It represents a fundamental shift in how we understand reproduction and species boundaries. These ants have effectively domesticated the genome of another species, co-opting Structor males into their life cycle and maintaining them as a lineage of clones within their own colonies. Think about what this means. Messor Ibericus has turned another species into a biological tool, a genetic resource that can be produced on demand. The Structor males are not invaders or parasites. They're manufactured by the Ibericus queens themselves, cloned and raised to serve a specific purpose. This system allows Messor Ibericus to expand into regions where Messor Structor does not naturally occur, breaking free from geographical constraints that would normally limit their range. It's an evolutionary masterstroke, a solution to a problem that most species could never overcome. But how does this bizarre system actually work? How do the queens clone these males? And how did such an unusual reproductive cycle evolve in the first place? The story begins in areas where the two species coexist, where Ibericus queens had access to Structor males for mating. Over time, Messor Ibericus lost the ability to produce worker ants independently, becoming dependent on sperm from another species to create their workforce. This phenomenon, known as obligate sperm parasitism, occurs in some harvester ant species. But there's a catch. Sperm parasitism usually limits a species' spread to areas where the host species exists. If you need another species' males to reproduce, you can't expand beyond their range. Messor Ibericus found a way around this limitation by producing Structor males within their own colonies. The Four Offspring. The reproductive system of Messor Ibericus produces 4 distinct types of offspring, each with a different genetic makeup and role within the colony. It's a complex dance of chromosomes, a carefully orchestrated system that defies conventional understanding. First, an unfertilized egg produces an Ibericus male through parthenogenesis, carrying only the maternal chromosomes. This is standard for many ant species, where males develop from unfertilized eggs and are essentially clones of their mothers. Second, an egg fertilized by an Ibericus male develops into a new Ibericus queen with 2 sets of chromosomes, 1 from each parent. This is normal sexual reproduction, the process that maintains genetic diversity within the species. Third, an egg fertilized by a Structor male can develop into a hybrid worker, retaining chromosomes from both parent species. These hybrids form the workforce of the colony, the laborers that gather food and maintain the nest. Fourth, and most remarkably, an egg fertilized by a Structor male can have the maternal chromosomes removed, leaving only the Structor DNA. This produces a clone of the male, though the exact mechanism remains unknown. It's as if the queen's egg becomes a vessel for copying the Structor genome. Researchers coined the term Xenoparity to describe this life cycle. Xeno meaning strange or alien, and parity meaning to produce or give birth. It's a fitting name for a process that seems more at home in science fiction than in the natural world. The Domesticated Clones. The clone Structor males living in Ibericus nests are not quite the same as their wild counterparts. They appear slightly different, particularly in hair density, with less coverage than wild-type Structor males. This may result from incompatibilities between their mismatched mitochondrial and nuclear genomes, or from the way they are reared in another species' nest. Researchers investigated whether these clonal males could break free from their domesticated state, whether they could interbreed with wild Structor males and escape their genetic prison. No evidence of such interbreeding was found. The clones are locked into their role, unable to escape their domestication. This raises profound questions about classification and identity. Should these isolated domesticated clones be considered a separate species? Humans have domesticated countless organisms, from dogs to wheat, creating forms that differ dramatically from their wild ancestors. Are these Structor clones any different? The relationship here is far more intimate than typical human-driven domestication, though. Both species are mutually dependent for reproduction, locked together in a biological embrace that neither can escape. The Ibericus queens need Structor males to produce workers, and the clone Structor males need Ibericus queens to reproduce at all. Genetic evidence currently supports grouping the cloned lineage with Messor Structor based on nuclear DNA, but this challenges our understanding of what a species actually is. These males appear as Structor in their nuclei, but carry Ibericus mitochondria, and they look physically different from standard Structor males. Are they a new hybrid species? Not exactly. True hybrids usually carry nuclear DNA from both parent species. These ants only have Structor DNA in their nuclei and must mate with an Ibericus queen to reproduce. They cannot propagate themselves independently. They rely entirely on another species to exist. The blurred line. This case highlights the complexity of life and the limitations of human labels for species. We like to draw clear lines, to place organisms into neat categories with defined boundaries. But nature doesn't respect our labels, doesn't care about our need for order and classification. The situation is comparable to endosymbiosis in cells. where eukaryotic cells incorporated free-living bacteria as mitochondria billions of years ago. Those bacteria lost their independence, becoming organelles that cannot survive outside their host cells. Similarly, these clonal structa males could be seen as organelles at a superorganism level, playing a crucial role within the colony system. At what point does a domesticated organism become something new? When does a species lose its identity and become part of another? These are not just academic questions. They strike at the heart of how we understand life itself. The discovery beautifully illustrates nature's complexity and an elegant evolutionary solution, allowing ants to expand beyond the range of another species. It's easy to take a human-centric view, to assume that our way of reproducing is the standard against which all others should be measured. But organisms like ants show just how diverse and creative life can be. In the world of insects, where colonies function as superorganisms and individuals sacrifice themselves for the greater good, the boundaries between self and other become blurred. The Ibericus queens are not stealing Structor DNA, they're incorporating it into their own reproductive system, creating a hybrid life cycle that transcends traditional species boundaries. The ants that clone their enemies remind us that nature is far stranger than we imagine. In the hidden world beneath our feet, evolution has crafted solutions that defy our understanding, blurring the lines between species and rewriting the rules of reproduction. This discovery is not just about ants. It's about the endless creativity of life itself. As we continue to explore the natural world, one truth becomes clear. We have only begun to scratch the surface.